Okay, so let's talk about a stimulus and depolarization of cardiac myocytes. So if a cell is going to be acted upon by a stimulus and it is of threshold intensity, the cell will respond to it. As the cell depolarizes from a polarized state, we will generate an action potential. As we depolarize, we become less negative and go towards 0 and then plus 30 millivolts, right? So we're going to go here. We're at about negative 90 millivolts here. As an action potential hits, we change those gradients, right? Which we'll go over in a second. All these ions are now exchanging. We're going from sodium's going to rush into the cell. We're going to go from a negative 90 to 0, so we're becoming less negative, and then we're going to probably peak up around positive 30 millivolts, right? So we're going to then positive at that point in time. Okay, so there's two ways we go from negative to positive. Okay, so here are the two ways. One, we increase the permeability to sodium, so we open up those sodium channels. Two, we increase the conductance of sodium, which will move sodium from the outside of the cell to the inside of the cell. So sodium will move in, and it will move in such a rush that will overwhelm the potassium that is moving out. So we will get to that point of plus 30 millivolts. Okay, so this triggers systole, stroke volume, ejection, all referring to the same thing. We're going to get into that with the myocardial mechanisms, right? So the difference here, when we talked about electrical potentials and neuromuscular physiology, it caused a muscle contraction. So you can flex and extend and lift a weight and lower a weight. Here, what is happening in the heart is that we want this to occur when we go from a negative state to a positive state, and we overcome that threshold. We are going to trigger systole. So where the blood is going to pump blood out of the heart, right? Out of the right and out of the left side of the heart. It's going to trigger stroke volume and all that. Okay, so we need these differences in ionic distribution across the membrane in order for it to respond to a stimulus. So then the heart can then contract and eject blood. That's the whole point and the heart's job, right? To receive and to eject blood. Okay, so in a resting person, this entire process we just talked about occurs about 70 times per minute. What does that correlate to? Our heart rate, right? So about 70 times per minute this occurs. While exercising, it can go upwards to, near about 190 times per minute, right? So again, let's reverse back to the same concept we talked about, how it's kind of wild in neuromuscular physiology about how these action potentials have to occur, move down, all of these gradients have to occur, sodium potassium exchanges, sends those electrical potential, the action potential down. So the same thing within the heart, this has to occur at least 70 times a minute, right? So we have to change that ionic gradient, those ionic gradients where sodium rushes in, we go up to plus 30, we eject blood out into the heart, and then we rest, everything goes back into place, calcium, potassium, sodium, all of that to get ready for the next stimulus, which is then going to eject blood back out into the heart. Okay, so remember now, after we depolarize, we have to repolarize, and this means reestablishing resting membrane potential. Okay, so we're putting back the ions where they came from. Sodium potassium pump works very fast. We close down those sodium channels and we open up the potassium channels, right? Because again, we want to open up those potassium channels because those naturally move out 100 potassium ions for every one sodium ion coming in. So we want to open up as many of those potassium channels as we can to further decrease that negativity in the heart. We want to go back to resting membrane potential, so we want to become more negative. Okay. So how quickly we repolarize determines how quickly we can respond to the next stimulus. So before we can actually respond to the next stimulus coming down, we need to put all of those ions back where they came from. This happens extremely rapidly, and again, we can't even appreciate the speed at which this occurs. We just take all of this for granted. Our heart pumps, all this stuff works, but the amount of time, the speed at which this occurs is extreme. So repolarization means a state of refractory, right? And we have two refractory periods, which we're going to go over. We have an absolute refractory period and a relative refractory period. Okay, so the absolute refractory period. This is a point in time of repolarization where no matter how strong a stimulus is, the cell will not respond. Not enough ionic correction has taken place in this period Okay, and this period usually begins as soon as you start the upstroke of depolarization. Now, the relative refractory period this is a point in time where enough ionic correction has taken place, and if the stimulus is of suprathreshold intensity, the cell will respond. The response will not be as strong as the first stimulus response, so the action potential won't be as high, but it will be slower. And you can still get a response if you hit it with an extremely high stimulus. It takes a lot more of a stimulus because, again, we are hyperpolarized. We are about 20 more units or millivolts away. So we need that extra power for stimulus in order to respond. Okay, so when we look at a cardiac action potential, we have it here, which we're going to go over the steps in just a minute. So when sodium rushes in, we become less negative, we go more positive, right? We eject systole, pump blood out, all that good stuff. And then we have to go back to a state of ionic correction and negativity in order for us to do that again. So when you look at this is the action potential graph, but then we can actually correlate it with down here with the electrocardiogram, right? So we have, which is missing over here, but we have the P wave where the atria are depolarizing, then the ventricles depolarize where we're going to eject the blood flow. And then now during this repolarization phase is actually where we can line up the relative refractory period, which is the T wave. The T wave is where the ventricles are repolarizing. Again, repolarizing meaning everything going back into place, right? So we can be ready for the next stimulus and the next ejection. However, so we can line that up right here. So this relative refractory period is where everything is going back into place, but just enough ionic correction has taken place that if we hit the heart or we hit these cells with a suprathreshold intensity stimulus, the cell will also respond. But the response won't be as great as that first response, and the original stimulus strength won't allow any reaction to occur. It has to be of suprathreshold intensity. All right, so if we zoom in and overlap these graphs a little bit closer, here we have the cardiac action potential, and then we have the T wave here from the ECG, which is the the repolarization of the ventricles. So if you look here, this ARP is the absolute refractory period, that not enough ionic correction has taken place, that even if we hit this cell with a very hard stimulus, it's not going to respond because not enough correction of sodium potassium has taken place. Now here is our relative refractory period, that enough ionic correction has taken place at this portion of the cardiac action potential, as well as we are repolarizing of the T wave, that enough has taken place that if we hit it with a suprathreshold intensity stimulus, that it will respond. But then now we go down here, and then now once we are back down here, we are back at resting membrane potential, and again down here, then we can hit the stimulus again and cause ejection and all that other good stuff in order for us to respond over here. We have usually the mechanical response back down here. Okay, so in the next video, we are going to go over the steps to the cardiac action potential.